Hi everybody. How are you guys? I hope everybody's doing well. Thank you for attending the IVM seminars. And today we have actually a guest lecturer, John Franck. Now, Frank spelled with CK, not with K. Very interesting. Um, so Frank has done what? You were a postdoc with Jack Fried, then you also did a postdoc with Songy, right?
Yeah, so I did grad school with Alex Pines, postdoc
Right.
with Songy, and then postdoc with Jack Fried. Yeah.
Right. And then in Songy's lab, you actually developed, I mean, you were pretty much the brainchild of all the ODMP was out there to detect water in all kinds of cavities and greatness. And I'm sure you're going to tell us all about it today. Is that about right?
Yeah.
So besides that, he's also a professor in Syracuse University, where he's leading a successful group. And I'm sure he will tell us all about what they're up to nowadays.
Very nice. Well, thank you very much for the introduction. Thank you guys for the invites too. And as as was introduced, we'll be talking about ODNP today, as well as I point out, and friends, which, not to do a spoiler, is basically ESR and deuterium relaxometry. Okay. So first, I like to introduce our kind of problem by pointing out that if you open any biochem textbook, just an undergrad biochem textbook, you'll typically see, and you turn to the chapter about the tertiary structure of proteins, you're typically going to see some type of statement that tells you that, okay, the salvation layer, or what in this talk I like to call the hydration layer, is very important, right? But then that's it, right? Like they sort of drop the topic, right, and leave you to make your own frequently kind of inaccurate assumptions about what is involved in this salvation layer, this hydration layer. Okay. The reason for that is that we know as physical chemists, right, that if you take, anything from an ion through to a very large hydrophobic solute, through to a solid with a structured pattern of hydrogen bonding, and you throw these into water, water is a fluctuating matrix of hydrogen bonds, right? And so it's going to respond in an often very complicated way, right, to the presence of these different solutes, okay? And that's a problem because going back to when people were first, first developing these really long simulations where they could actually watch with molecular dynamics what happens, for example, when you have peptides that aggregate in solution or other such fundamental measurements or other such fundamental processes, I should say. What they found is that they could go in and use some nice stat mech to pull out what the contribution of the hydration water was. And what they found is that the contribution of the hydration water is that it was contributing a chemical potential that was driving this process forward that was at least as big as, and at certain points in the process, much larger than the types of energies that are coming from the peptide backbone itself, right? So this is something very complicated, but this is also something that we absolutely want to address, right? And so how are we going to do that, right? And of course, we're going to use magnetic resonance to do that. But to point out what it is exactly that we're measuring, I like to do a little kind of fun thought experiment, okay? And this is my former graduate student, Alec. He has graduated last fall and now works at Brooker. And so he's a good sport. And so I dragged him outside one day and I said, okay, can you balance a penny on your fingertip, right? And so we have, international people in this crowd. It's about a, centimeter, right? And so I asked him, you balance a penny on your fingertip, and I took a picture of him like so, right? And that provides us with one sense of scale. And for another sense of scale, this is a rendering of an E. coli. And if we shrunk Alec down so that he was this big, right? And we asked him to poke around with his fingertip, he would find that on certain surfaces, water behaved pretty much as he would expect from his bulk intuition, right? Water just behaves like water. But on many other surfaces, he would find that it seemed viscous or sticky, right? On other surfaces, he would actually press and his fingertip would feel hot because, there would be enthalpy released into his fingertip. And in other places, his fingertip would feel cold, right? And of course, these things are all very important because this gives us sort of an head picture of what happens at this nanoscale, right? And the types of energies that are involved. And these types of energies are exactly what proteins and proteins and DNA and polymers and all these types of things experience, right? And it really highlights the fact that there is a type of structure, right, to water at the nanoscale, right? So there's different coloration here. We're going to be looking at translational mobility as sort of a handle, as a measure of how things are different, as well as rotational mobility, okay? But I want to point out that that's just one thing that's changing right? That the water at these differently colored sites is different in a whole host of ways. It has a different residual entropy. There's a different activation energy to move it out of the way. All sorts of things are different, right? And really, right now, what's happening in biochemistry is that people are kind of using as sort of a crutch, right? The idea of AI to basically use pattern matching to try and figure out, oh, this missing piece of energy, right? This is one of the pieces of energy that's missing that we don't have a way of measuring, right? Or we didn't have a way of measuring, I should say, right? But what's important to know is that this is really difficult, right? Because at this length scale, there's our nanometer again, right? At this length scale, this water is zooming in and out of this frame on a sub-nanosecond time scale. So even though it's behaving differently at these different sites, individual water molecules are zooming through this frame at a very, very fast time scale. Okay. And I wanted, this is my reminder to point out to you that I like to use biochemistry to provide a sense of context and scale, but this is very much relevant to all types of macromolecules, synthetic polymers, all sorts of things like this as well, right? As well as other types of solvents, as well as solvents that, have a structure similar to the way that water does. Okay. And of course, the tool that we're going to be using to address this is ODNP, or Overhauser Dynamic Nuclear Polarization. Okay. And as I like to tell my students, right, Overhauser DNP is in a lot of ways a completely different thing than solid state DNP, right? And so I like to talk about, okay, what is it exactly that we're looking at and what type of information is it telling us? Okay. And to understand that, here's a water, right? And of course, it has a magnetic moment, right? That's really tiny. And of course, we're going to make things more interesting by introducing an electron spin, typically in the form of tempo group, right? So this could be like an MTSL attached to a protein. It could be a tempo group attached to a lipid. We synthesize small molecules that, have tempo functionalized with different types of things, right? Other people in the audience certainly do this as well. And so what we're going to do is that we're going to go ahead and use microwaves to saturate our electron spin. Okay. Now, the thing is that under the conditions that we're using, right, without molecular motion, that would just be the end of the story, right? I have an electron spin, it's saturated, I have some proton spins hanging about, right? But things become interesting when we bring molecular motion into the picture, because molecular motion very specifically opens up cross-relaxation transitions that allow us to transfer polarization from that pretty big electron spin with that big magnetic moment onto the proton spin, which was previously tiny, okay? And so this encodes the water molecules in our solution with information very specifically about what's happening within 1 nanometer of this spin label, right? And specifically, if the water's not moving, we're not going to see much transfer. If the water is moving, we're going to see a lot of transfer, okay? And so I like to talk in a little bit more, kind of precisely to give you an idea, especially with the magnetic resonance crowd, of what it is that we're actually seeing here, okay? So here's our water zooming through the field of our spin label, okay? And that is going to introduce, right? Quantum mechanics tells us that we can get a transition, such as the transition we need to exchange polarization between our electron spin and our proton spin, if we have a time-dependent interaction that is fluctuating at the right frequency, okay? So our time-dependent interaction comes from the fact that these guys both have magnetic moments, and the molecular motion is moving them relative to each other. So I have a fluctuation of my spin-spin dipole interaction. But of course, we know that that has to happen at the right frequency as well. So we need to look at how is that Fourier transformed and averaged over the ensemble. This is what we call our spectral density function, right? And by the way, of course, I'm giving you a light touch here, right? I mean, this is, sort of when I started my independent career, this is a nice review that I wrote together with Song-Yi explaining this whole thing, okay? And so when we have slow motion, right, we Fourier transform, and those interactions are not fluctuating fast enough to excite this flip-flop transition, right? So this flip-flop transition that we need to give us DNP is out here at 10 gigahertz, right? Because our electron spin is basically dominating the frequency. So if motion isn't happening at tens of picoseconds, it's not moving fast enough to excite this transition out here at 10 gigahertz, right? So our NMR is not going to get that encoding that we were talking about before, right? On the other hand, if it's fluctuating quickly, then in the frequency domain that reaches out to 10 gigahertz, it excites that cross-relaxation transition, and we get this huge transfer of polarization from our electron spin onto our proton spin. And so we can look at these nanometer scale sites inside this huge bulk water solvent and see a distinct signature from that, okay? So this is the basic idea, how this works and actually, again, and here's another reminder that I actually forgot about this time. This is for me to remind you that a really important thing here is that even though what we're looking at is NMR signal, what we're fundamentally limited by in practice, right, are sort of the experimental requirements of CW-ESR, right? So this means we're doing this in liquid state. We can work with very viscous things. We can work with very large molecules, right? So we can do things like transmembrane proteins. We could even embed them in a liquid bilayer. We can look at very, gummy, viscous types of solutions, right? And all of this works fine, okay? And the way that this works is that, this is, as I've been indicating, basically a double resonance technique, right? So we can sweep our static field, right, across our ESR transition. You see 3 spikes there from our 3 hyperfine transitions. And what we typically do is that we sit on top of one of these, the maximum of one of these transitions. We crank up our microwave power, right? And there's that encoding of our NMR signal. It flips upside down and it enhances, okay? If we have a small concentration of spin labels, we'll get maybe an enhancement of 2 or 3, and we're fine with that, right? Because what we're interested in is the information that this gives us, okay? That red dot that I was pointing to before, I did not point out, right? We call this K sigma, right, for cross-relaxation, stealing from the nomenclature of NOE, okay? And I'm pointing out that we do need an automated measurement here because one of the things we need to do is that we have subtle changes in our NMR background relaxation that we need to account for, okay? And as a postdoc, as Tomas was saying, this was one of the things that we figured out is how you account for, those subtle changes in background relaxation to pull out an accurate measurement of your dynamics. And specifically, I really am going to highlight the interpretation of this number, as this is the number of fast-moving water molecules that we're seeing at this particular site where we have our spin label, okay? So if we have more fast-moving water molecules, we get more cross-relaxation. If we have less, or they're moving more slowly, we get less cross-relaxation, okay? Now, when I started my independent career, right, there were 3 main questions that I wanted to address, okay? And I'm going to walk you through, in the middle part of this talk, I'm going to walk you through these 3 main questions, and then we'll focus in particular on one particular study towards the end of this talk. The first of these is access, right? How do we make automated measurements with a simple and clear interpretation? Okay, so you've already seen some of this with my introduction, right? I also want to point out, right, that this is a measurement. We like to do this because we like taking nice CW-ESR measurements. We're doing this on a commercial Brooker EPR spectrometer, and then all of our NMR stuff is home built, and then we have a nice custom high power microwave amplifier from Bridge 12, okay? And I want to emphasize that this is happening at a field, though, of 0.35 Tesla and with a very small sample volume, right? So this is really something that is on a trajectory to be something that you can just have in a biochem lab, right? And if people are able to do the biochemistry to like attach spin labels at different sites on the surface, they can really just kind of go ahead and crank through these measurements, right? This is really the trajectory that I see this going, right? And for implementing this now, right, one of the things that really is something that needs to be done is that you have various different modules, right? And so our ESR spectrometer, microwave source, our NMR spectrometer, right? And all these things need to be integrated together, right? And what we've found is that Python forms a very nice glue where we can write, we have a whole bunch of home-built libraries that we use both for data acquisition and processing that really make this whole thing relatively seamless, okay? And this here, I'm just showing you that we have libraries that like sort of compress what it would otherwise be relatively complicated code into relatively simple, into relatively simple few lines of code, okay? The other point of, under having a clear interpretation is that we're tackling some temperature dependent studies as well, okay? And these are preliminary measurements, but this is what I'm showing you here is reproducible, where you see reproducibly that as I change the temperature, specifically as I cool down a solution of water with just a small tempo label inside of it, I see that this cross relaxation rises to a distinct maximum, right? At about 4C or something like this, okay? And we like to call this a dynamic resonance, right? Because what's happening there is that actually you're getting maximal cross relaxation because the time that it takes the water to move through the field of the spin label is matching your resonance frequency, it's matching the characteristic frequency of that transition. And in fact, we can go to Marina Benatti some years back, did some nice field cycling relaxometry studies where she has a model that is experimentally verified for the entire spectral density function for some of these small molecule spin labels. And actually, we can extend that model, and we find that, yes, indeed, we do see that we're supposed to see this maximum, and it's supposed to happen at about the spot that we're seeing it, right? And so this is actually really exciting. Although abstract, this is pretty exciting because it's actually highlighting the fact that this is really a measurement that could tell you about dynamics, right? You can see this dynamic resonance that's unambiguously the time scale that your list tells us at this temperature, right, we're seeing a correlation time that is matched to the inverse of that 10 gigahertz frequency, right? So that's pretty cool. The other thing is the second question is applicability, right? Is this a good technique for both patches and pockets of water in complex environments? As you might have gathered, I'm more of a physical chemist, right? And so I really wanted to see that it was very practical for us to go ahead and, take a couple nice biochemical systems, spin label them at different sites and see what's going on, right? And so on the left here, I'm showing you this is proterodopsin. This is a transmembrane protein, right? And here I'm illustrating this is a Rosetta model of our library of spin labels. Each different color is where a spin label is where a differently attached spin label would go, right? And sort of the distribution of positions we believe that it would exhibit at room temperature, okay? And so you can see we have ones at the surface as well as going down into either the lipid bilayer or the proteomycel, depending on how you've prepared your samples, okay? And what we found, what we knew from before, right, from studies out of Song-Yi's lab, is that there is quite a bit of variation in what's going on these surface sites, right? But what we're doing now is that we're going down into the lipid bilayer, and what we're actually finding that's interesting is that the variation, of course, there's less fast-moving water down inside this lipid bilayer, but not as much less as you might kind of naively think from just this idea that, okay, I have water, then I have lipid or surfactant, and then I have water again when I have a transmembrane protein, right? So there really are distinct pockets of water that we're seeing open up next to the protein, right, that we can pick up with DNP, right? And so that's pretty cool. We also find out interesting things that, of course, when you change out as you kind of might expect, given that if you change out your surfactant, right, you're going to see pretty dramatic changes in how much water you're actually seeing next to the protein here. We're also seeing that actually due to the tumbling of proteomycels, right, the NMR relaxation of these guys actually responds pretty dramatically to very small changes in temperature, right? And that's just something that wasn't really known before, okay? The other type of system that we're attacking, right, are not these messy transmembrane systems, but globular systems, right? And so here we're looking at, this is KRAS, this is a signaling protein that is important to cancer pathways. What we care about is that this is a signaling protein, right? And it engages, and even though it's pretty much like sort of a weird, say, football, right? It engages in these extended interactions with binding partners, right? So large parts of its surface, bind to other things when it's doing its job, basically, okay? And so what we have an opportunity to do here is that we wanted to see, can we go in and attach some spin labels and see whether or not we see distinct patches where we have different types of water dynamics at different parts of the surface, right? And we do think that this is we are seeing this, right? So here you can see, here's some resonance. So the gray are where we have not attached spin labels. You see a red patch here where we have faster water dynamics. You see a blue, and slightly intermediate colored patch here where we have slower dynamics, right? So we're actually seeing for the first time residues that are near each other, where when we attach spin labels at those sites, we're reporting on a similar dynamics because they're all looking towards the same patch of water, right? And so this is neat. And so this really adds credence to this idea that your protein is surrounded by this hydration layer that has a distinct fingerprint, and that distinct fingerprint helps control its interactions, its binding interactions with other proteins and things like this, right? And this could be particularly important to signaling proteins, okay? Okay, and then the third question, right, is a question actually that I had gotten a bit before, is that, okay, well, so what can you say about, doing these types of measurements? What can you say about making predictions as to what the water is going to be doing at different points, right? And taking a cue from molecular dynamics, there are many papers that sort of talk about this. This is one that I just like, I know these people and they state this very clearly, right? Is this idea that you might think naively that what the water is doing is going to be controlled by whether the surface at that particular point is hydrophilic or hydrophobic, right? And this is a really common assumption that is made in biochemistry, right? But molecular dynamics actually gives us pretty strong indication that the shape of the surface, whether you have a pocket or a flat surface, right, what is the curvature at that point, actually has an outsized influence on how the water behaves, and of course, the types of energies that can be harvested from the water during interactions, okay? And for this, we're going to focus in on a nice model system that we can use to study water that's confined to different volumes, or if you want to think of it differently, water that is experiencing different curvatures, right? And this is the reverse micelle system. Here we have a pocket of about nanoscale water, right? It is surrounded by surfactant, and then it is suspended in an apolar organic solvent, okay? So this is an interesting system. There's our nanoscale alec again, right? These are our reverse micelles. And you'll notice that I have a black background here because there's a lot of organic solvent around that we don't want to hear from, that we're not interested in, okay? And so this is because we have this tiny little sample volume, this is actually challenging for us because this is a very low proton type of measurement, right? So we have relatively little water that we're looking at, okay? And so to really understand what's going on here, we're going to do two things. One is that we're going to reach out across the table to other magnetic resonance measurements and pair this with deuterium NMR. And the other thing that we're going to do is that we're going to improve our ODNP data analysis, okay? And this latter one is covered in this editor's pick article that we have in JCP. And because it's a little bit more of a magnetic resonance crowd, I wanted to take a minute or two to just talk about what's going on there, because that is pretty relevant to a wide range, actually, of different magnetic resonance types of measurements, okay? And this really hits at the idea that, well, okay, we're using spin echoes to detect our NMR signal, right? And everyone, right, even without thinking about it, is using a technique where you're going to pick out exactly the signal that you're interested in. You're going to clean up your signal by using phase cycling, right? You're going to pull out a particular coherence transfer pathway, okay? The issue is that when we do this in a room temperature electromagnet at 0.35 Tesla, it turns out that phase cycling does not work as you might expect at first, right? And so in our particular case, we were really motivated to look at the science behind this in a different way, okay? And we do this with two different steps, right? So step one is that we take our phase cycling of our pulses, right? So basically we have a series of transients in a phase cycle. We separate that into a new dimension, right? So in our case, a third dimension, we have, frequency, microwave power, and now we have this phase cycling dimension, right, for our data. And the other thing is that we need to be able to visualize data without necessarily phase correcting it first, right? And so we use this special color scheme where color represents the phase of our signal, right? And the intensity basically is the magnitude, okay? And that allows us to draw pretty pictures that look like this, right? So this is the raw data from a DNP experiment, right? So these individual blocks are resonance frequency, microwave power, okay? And then these three stacked blocks that you're seeing, that's that extra dimension that stores our phase cycle, our phase cycled signal. And you'll see that it's changing color because we're cycling the phase of the pulses, right? And of course, going back, decades, Bodenhausen and Ernst, the physics that they tell us essentially says that if you take these guys and you Fourier transform along this extra dimension that we've introduced, right, then this goes from being phase cycles to being coherence transfer pathways, okay? And so all of our signal ideally gets dumped into the coherence transfer pathway, where we expect it to see, and most of it does. But you'll notice that in our case, we have this extra smudging here, right, that is we like to call phase cycling noise, right? This comes from the imperfections in our system, and specifically you'll notice that with each individual transient our resonance frequency is bouncing around, right? Because our Brucker electromagnet, just the way it's designed, does not control field that accurately, okay? And so, of course, we can, what we're doing here is that red is real. So we're basically just phase correcting our signal. And, we actually have this isn't totally standard. We have some custom algorithms to do this nicely for a spin echo. I didn't really mean to tell you that. But then the real part that I would highlight from this, right, is that looking in the signal this way, what you can actually do is that you can go ahead and now that since I have all the information about all the data that was acquired, I can now develop algorithms, for example, to clean up that resonance frequency shift to just shift these guys so that they're all aligned, even in the presence of phase cycling, right? And I get nice, clean signal that's all aligned, right? And also you'll see too that I've really eliminated most of that phase cycling noise by doing this, right? So this gives you a new perspective. And also too, as part of that, there's, new procedures that come out of it. Okay. And so that's neat, right? And then the other thing I really wanted to emphasize is not just that you can do things like this with this, right? But also that if we go ahead and look at challenging systems like these reverse micelles, we see the value of the visualization technique itself, right? And that is this, right? So this is the signal that's acquired from these reverse micelles. So the previous signal, I'm sorry, I should point out, is just a normal aqueous sample. Okay, it's a spin labeled aqueous sample. Here we have reverse micelles where we have very, very little protons, right? And if I just looked at a 1D signal, it's very hard to see if I even have signal there, right? And actually my graduate students had run this and told me, I don't know what to do because I don't see signal, right? And it actually turns out that if we look at it this way, you see this distinct signature. We see this red band. That's our coherent signal. So this, in this case, is just multiple transients or multiple scans that were acquired one after another, right? And we see this little red band that we really kind of have a lot of trouble seeing in a standard way. And we can say, hey, that's my signal. And then we can say, hey, let's go ahead and apply this correlation alignment algorithm that we just talked about in the previous slide. And lo and behold, just by shifting these guys so that they line up in field, we can dramatically improve our signal to noise, okay? This is not, it's not weird, by the way, that's because we're acquiring an echo. And so we're getting an improvement in signal to noise that is not, this is not denoising, right? We're not doing anything funky to our signal. All we're doing is aligning the resonance frequencies for all the transients, right? And so that's pretty cool, right? So now we can go ahead and we can attack this reverse micelle system. And we can say, hey, looking at the literature, it's pretty known that this confined water is going to behave differently. What's really nice about this system is that I have pockets of water where I can leave the surface chemistry alone, use the same surfactant over and over again, but by changing the molar ratio of the water molecules to the surfactant, I can change the size of this pocket of water, okay? That's basically what I said there. And then Alec did a bit of work to find and synthesize a nice spin label that really sits in the middle, and to verify that it sits in the middle better than other types of spin labels by using just standard NMR-PRE measurements, which I'm not really getting into here. Right. And we're also interested in this, not just as a model system, but because there's a really nice method for looking at variations in the properties of water. But what you have to do is that this is by the Wan group, right? And what you have to do is that you have to take the protein and you have to take a protein and put it inside of a reverse micelle bag in order to make this measurement, right? And so we're interested in this not just as a nice controllable system for understanding confined water, but also to specifically ask the question of, when you do something like this with a protein, is this the hydration layer that you actually would get in the bulk, or are you changing it by sort of confining it in this way? Okay. And so what Alec did is that he went to the literature and he found, there actually is a way to figure out, what my DNP enhancement is. They hadn't done the full dynamic analysis for this, but what they had done, in which he repeated, which is that I can go ahead and I can take this measurement, right? And when I do DNP, right, I get this type of increase in my signal that I expect, right? But this is coming from both your water, and there's a significant amount of surfactant protons there as well, right? So what he does is that he substitutes the water with D2O to get just surfactant only, subtracts the two so that he can see what's coming from the water, right? What's neat is that as you go through and change the size of the, of this water bag, right, you see that, all of a sudden, right, at a few nanometers, right, the DNP enhancement really just jumps, okay? And so you're seeing that for small reverse micelles, I get actually, if you zoom in there, right, you are seeing this kind of asymptotic dependence that you expect, right? But you're not really seeing much cross-polarization, right? You're basically seeing essentially no cross-polarization. And then as you increase the size of the water can now move in a way that is more like in the bulk, which is faster, right? There's more hydrogen bonds for it to exchange and move things around, right? And then all of a sudden it's translating at a much better rate, and I can get much better DNP, right? And so doing our kind of analysis where we actually pull out the actual cross-relaxation measurement from this, that's sort of the raw data, right? We get a dependence that looks like this, right? What we can actually go ahead and compare this, right, to rotational. So this is measuring our, translational mobility. Okay, and we could go ahead and compare this to rotational mobility by doing a companion study where we use deuterium relaxation. This is on a higher field magnet, right? And we actually came up with a new little technique here that lets us look at the rotational mobility, the correlation between the rotational mobility and the hydrogen bonding strength, right? And you can get this because, as has been known for a while, D2O relaxation is related to your rotational mobility, right? And then also your chemical shift is related to your hydrogen bonding strength, right? So just using some modern processing techniques, we can get a nice correlated measurement between these two, right? And you see that, okay, over here, I see this kink in my translational dynamics, right? This, by the way, is still a decent amount of water molecules. This is like 730 water molecules, right? Where my translational dynamics seems to kind of shut off, okay? I see at the same water loading, water loading is the size of the reverse micelle. So at the same size reverse micelle over here, right? Water loading is actually the ratio, which I had on a previous slide, but I should say these numbers here, this water loading is the ratio of the moles of water to moles of surfactant, right? And so at the same size reverse micelle over here, I see this kink in this correlated, measurement, right? So that's kind of neat. And then the other thing that I see is that, okay, if I were to extrapolate this, right, and say, what do I need to do? So it actually turns out that here we're still looking at, about 10 nanometer size pools of water. If I were to extrapolate this, and what we're seeing is that the water here is moving at about a fifth of what it is in the bulk, right? So if I were to extrapolate this and ask, okay, how big would this water pool need to be for me to see bulk-like dynamics, right? I would need a pool of water about 10 nanometers, right? So that's 27,000 water molecules, right? That's how much collective, cooperation between the waters needed to give us this bulk-like motion, right? And similarly, right, if we look at the correlated plot over here for the rotational motion and hydrogen bonding, right, that would correspond to a point on this plot at about here, okay? And so, yeah, and so what we're seeing is that, water is really dramatically affected by any type of pocket, any type of confinement, right? It's really affecting its properties, as we can see either from translational motion or through correlated rotational mobility hydrogen bonding strength, okay? So we've learned some things, and I still have a few slides. Is that okay if I have a few slides still, I think? Okay. So we've learned some things, right, from these DNP measurements, which is really neat, right? One is that, magnetic resonance observes some nanosecond dynamics in the frequency domain, right? The other is that nanometer confinement, really, I like to use this phrase, which I don't know if it's fair or not, it quenches the translational motion, right? So you take something, put it inside of a small pocket, translational motion is really, quite dramatically quenched, okay? It is possible, and this is a very applicable method. It's possible to absorb patches of different kinds of water on different types of proteins, whether it's a transmembrane protein or a signaling protein, right? And then also another thing that we've learned, and I'm going to show you a little piece from this here too, is that by pairing DNP and even just CW ESR, we can actually learn quite a lot about the solvent environment of transmembrane proteins, right? And this being an IES, sort of adjacent talk, right? I wanted to talk to you a little bit about this piece, right? And the basic idea is that what we're doing here is that we've learned that we can, with a modern Bruker ESR spectrometer, you can use overmodulation reconstruction pretty routinely. This is a proteorhodopsin, a spin labeled proteorhodopsin sample, right? This is showing you, okay, if I get a nice spectrum, if I'm willing to wait, like I forget if it's like 20 minutes, half an hour, or something like this, I get a nice spectrum, right? And then I say, well, okay, that's nice, but I want to do this for a bunch of mutants and stuff, so I don't want to wait that long, right? So let's use less time, and of course you don't get a nice spectrum, okay? But then we can say, okay, well, we know from the literature that I can overmodulate, right? I can acquire all of my harmonics, right? And then I can reconstruct what the signal should look like, right? And you can see here, this is much faster than this guy, right? And so you can see here that, I do reconstruct, I clean this up, right? And I clean it up in a way that by comparing to the long acquisition is quite accurate, actually, right? Now, of course, overmodulation reconstruction is something that's out there in the literature. What we're doing here specifically, though, that's really neat is that we have a standard regularization framework where we don't have any filters or anything like this that we need. This just pops out directly. And this is self-calibrating, right? So you don't need to do any type of calibration. Okay. And so we're really, at the point now. And so now I've told you basically all the, if you know enough about regularization, I basically told you all of our and pseudomodulation. I've told you all of our secrets. And so now I have to finish writing this paper. And so but really, we're really at the point where we can routinely use our E500 in overmodulated mode to acquire accurate ESR spectra like routinely, right? And so that's pretty neat. Okay. And I want to end this just by making a plug for two things, right? Number one is that if you are a programming person, we have a really nice in-house Python library that we use, right? This is uses a little bit more advanced programming concepts, right? But if you're really especially if you're into using Python for data processing, shoot me an email, talk to me. We would be more than happy to hook you up with this, get you to test this out for your own needs, right? We think that this is really neat, right? The other thing I'm making a plug for as well, though, is that if you just you want to run ODNP and you want to be able to process the data, right? This is a collaboration that's led by Bridge 12, including myself, as well as some folks from Songy Han's lab, right? This DNP lab is really designed to be an easily deployable, very sort of MATLAB-like interface, right? To just sort of do routine ODNP data processing, right? And so this is a really nice thing too. And again, if you shoot me an email, I'm happy to hook you up with this as well. You don't have to go the full, fancy Python route. Okay. That said, I'd like to thank all the people who did the work. I'd like to thank, NSF for career funding. I'd like to thank, startup funding from SU. And I would also add, of course, have any undergrads who are looking for good, graduate positions, send them our way. I actually, maybe I shouldn't, but I should say that, we may soon have a postdoc opening as well. And so, yeah, definitely send people our way to do cool science. So that's that. And with that, I'll take any questions if we have time for that. So, yeah.
All right, that's awesome. Thank you, John. You certainly did not disappoint as always. With that being said, we come to the most exciting part. Do you guys have any questions? I think that is really, if you do have a question, feel free to just speak up, or also you can type it into the chat and I can ask a question for you. Ask away for that pretty exciting stuff. While we're waiting, I'm just going to take the spot anyways. Hey, so in this case, you're looking at the water, obviously, that makes sense. And you're having your paramagnetic obviously attached to a protein or whatsoever. So is it possible that instead of actually looking for water, I mean, in theory, I can look for any either soluble molecule which can collide with the surface, but more importantly, I could also look for sidechain dynamics, which are in close proximity, because 10 angstrom is a pretty far distance. So if I put my label there, then I can get all the dynamics of a bunch of sidechains so long everything else is deuterated or specifically labeled. Is that correct?
Yeah, I mean, honestly, though, yes, right. But actually, I guess yes, right. I shouldn't say, because I'm used to working at low field, right? And so, to be honest, a big part of what we're relying on in our case, right, is that we have these solvent molecules that basically over the course of an NMR relaxation time, pretty much all of the solvent molecules in our solution have a chance to interact with a spin label, right? And so really it's a huge effect because of that, right? But on the other hand, I guess if you were doing DNP, say 95 gigahertz at higher field, right? Yeah, I mean, the idea that yes, you can apply this sidechain dynamics is not out of the realm of possibility, right? Yeah.
Yeah. Right. And so right now,
I would say too, right? I mean, what I would say, what I think more about, but to be honest, right, there's some challenges with that. What I think more about are things like, okay, moving to different solvent systems for sure, right?
Yeah.
And I think that would be a very interesting thing, right? And one of the things that we've is that now there is some capability to resolve chemical shifts at lower field, right? And
I mean, is that the reason why? I mean, I'm just curious right now. Maybe I didn't understand it fully. Right. The reverse Meister system, what you're using, I mean, partly is to get that to move faster, right? Isn't that the reason why? Partly though.
What's to get it to move faster? Sorry.
You're using the reverse Meister reverse Meister system, right?
Mm-hmm.
And because this one is an organic solvent, and then it's a pretty much reverse micelle. So the waters and everything should move faster inside the micelle. Is that right? Or is
No,
it
they move.
only
I mean, we see that they move slower, right?
right?
They do use this. Maybe what you're thinking about is that they do use this in like protein spectroscopy.
Yes.
Because the reverse micelle, you can put the reverse micelle in a very low viscosity solvent and have it tumble very quickly, right?
That's yeah.
Yeah,
Right.
yes, yeah, you could do that. Yeah, but
Right.
that's not why we're using it. Yeah.
Okay.
We're using it just to look at the confinement, right? Like how big it, I mean, so how does the translational dynamics of water change as you change the size of this water pool, right?
Right.
What you see is that as the water pool becomes slower, the water basically just stops moving, right?
Right. So did you ever come across like a water, like some waters which are just almost like stationary, like which are actually
I mean,
very
so this was actually, so honestly, that is what we're seeing with very low water loading reverse micelles, right?
uh-huh.
And honestly, like this is not something that we had really seen before, right?
Yeah.
I mean, this is really, I mean, so from my perspective, to some extent, like, oh, it's a neat picture in terms of like the collective dynamics of water, right? It's actually really neat that way, right? But there's also like a flip side to it, that this is also demonstrating sort of the range of the DNP technique, that you can actually also use this to look at really, really slow moving waters as well, right?
Right.
Especially if you have the deuterium relaxometry to sort of back that
Right.
up, right? Yeah.
Right. Actually, Boris is asking a question.
Uh-huh.
Would it be informative to look at the difference? To look at the difference in water dynamics and DNP enhancement, encapsulating the water molecule inside the carbon nanotubes or fullerenes?
Yeah, absolutely. Right. You know, so
I
honestly,
agree.
as a postdoc, absolutely, right? Because also these are really interesting systems where people believe that they observe very interesting like water dynamics, right?
Yeah.
Like actually one of the, I think of the classic study from like a few years back, where like, for example, people took like graphene sheets and they did like an X-ray scattering study, and you see these like very bizarre fluctuations and the water density that like David Chandler and these guys predicted, right? And so yes, those types of systems are very interesting. And you know what I'm the reason why we haven't, quite honestly, is that like getting one of these with a good spin label is more of a synthetic challenge than than I want to deal with personally, right? So if you have such a system, definitely come talk to us, right? Because that would be really neat. Yeah. I mean, honestly, like I've really thought about, like, for example, like the fullerenes with the nitrogen inside is a very nice it's an easy to saturate system, right?
Yeah.
That would be a really neat system to study, right? But we don't have easy access to those. So yeah.
Right. How about Jan? Do you want to speak up?
Yeah. Thanks, John, for the nice talk. I'm always a little bit concerned for how one can deconvolve the number of water molecules and their mobility. And I mean, you talked about the membrane protein, and obviously, as you go down in the membrane, you're less water molecule. You have less water molecule, and as far as I understand, K sigma contain both the number and the time scale.
Yes, it does, right? And so the traditional way of doing that, right? So there's two answers to this, right? So the traditional way of doing that is by like what we had done in Sung Yi's lab, is that use this other number, this K low, right? And you sort of, you compare so that's basically your spectral density down at low frequency. And sort of by comparing these, if you can, the big caveat being, if you can assume a particular model, right, then you can actually turn this into a timescale, right? But this is actually, the two answers to this are, one of which is already answered, right? Which is, that's actually what I showed you in the beginning, is that with these temperature dependent studies, right, if you can find this maximum, right, this what I call like the dynamic resonance, right, like if you can find the maximum as a function of temperature, that is the temperature at which your correlation time, right, is the inverse of your characteristic frequency, right? Yeah, I mean, and so that maximum is entirely a dynamic effect, right? And actually, I did this, I can pull these up, so this one, just give me 1 second, because I want to point this out, because this is really important, I think. Here we go. Boom. This guy. Okay. Second. Oh, having a Zoom issue. There we go. Okay, so this maximum here, right, is governed by dynamics, right? That's all, right? That has nothing to do with number of water molecules, right? And actually, the neat part is that actually you can see, what I'm not showing you here is that we can actually, essentially, we can watch K-low at the same time, and K-low is like entirely monotonic, right? This maximum only happens in the K-sigma, and it happens there because what's happening, right, is that, you know, here the water molecule is moving faster than the correlation time is faster than the inverse of the characteristic frequency. Here it is exactly matched, you get maximal transfer, and then here it's slower, right? Now with proteins and stuff, we're on this side, right, most of the time, right? And so this is why actually the other half of my answer is that this is why the other thing that we're doing simultaneously is that we are building a lower frequency system, right? Because in a lower frequency system, this maximum happens, you know, down at slower motion, right? And so then again, we can unambiguously, you know, see what's happening, say, on the surface of a protein, you know, unambiguously at what temperature, say, do we match the inverse of the resonance frequency at S band? Right.
Is that
sorry, that was a very long answer, but yes.
No, that's interesting. But is that something you've actually done in practice, like following this temperature, the optimal or maximum temperature, or the temperature for the maximum K sigma? As a
no. So what I'm saying is that, yeah, so for right now, what I can tell you is that we do see this maximum. This maximum does match where we would expect it to be based on Benatti's field cycling relaxometry measurements, right?
Okay.
Yeah, that's where we're at, right? We want to do this on the surface of a protein at lower frequency, right? You know,
yeah, I see.
Genuinely 6 months, a year, something like this, right? Like, yeah, maybe I shouldn't say that, but yeah.
Cool. Nice. Thanks.
Great.
No, but yeah, we're very. I got excited by the question because we're very interested in that question too. Yeah.
Yeah, no, I think that's. I mean, that's
it's an important question.
General
right?
concern that anytime you confine your water, it's very likely that you also decrease the number of water.
Yeah.
In addition to changing the mobility. So cool. Well, thanks.
So actually, Boris has another question. In this confined geometry, does bulk temperature deviate from the local temperature? So pretty much, what does temperature do? How does temperature react in the confinement space, or how diffusion comes in?
Yeah, I mean, I think that he might be alluding to literature that I am not familiar with, if I'm being honest, right? But like, I would say that, you know, kind of the baseline assumption is that everything is at equilibrium, right? And because these are equilibrium measurements, right? And so everything is at equilibrium, and so it's all sort of by definition at the same temperature, right? This to me sounds like maybe there are papers where they talk about defining a local temperature, you know, in an equilibrium system, but yeah. And I would be interested to know if that's the case, but yeah.
Great. Graham, do you want to speak up?
Yeah, great talk. Very, very interesting. Two questions. The first is following up on your, I guess the last question. How low in frequency do you need to realistically go to see significant differences? And secondly, what is the smallest delta that you can realistically measure in terms of differences in the DMP? Or is relevant is perhaps.
Yeah, no, so the so honestly, I would say, so the differences in the DNP, right? Like I would say, like, you know, I think realistically, because honestly, a big part of, you know, realistically, what are the differences that you can measure? Honestly, I would say for the most part is actually limited more so by, like, you know, sample preparation, especially when you're dealing with protein systems and stuff like this. It's more limited by, like, oh, have you got your sample preparation exactly the same between different repeats, right? But I would say about, you know, a few percent, right, you know, is the difference that we see. I mean, is the differences that we can sort of think we can resolve in this cross relaxation rate. and sorry, your other question was how low in frequency, and the answer to that is basically we are going down about an order of magnitude is what, like when I say we're going down, lower in frequency, like what I mean by that is like it's like an order of magnitude, right? So we're going down into like S-band, right? Yeah.
And so is that feeding a separate spectrometer, or is that an S-band system built into your?
Yeah, no, so in this case, we're basically just, this is actually what's nice about the setup, right? Is that because we have everything set up very modular, right? And like the NMR part is like home built, right? We're right now not immediately planning on doing S-band ESR, right? We're just going to read our ESR out through the DNP, right? And we're getting another microwave power source from bridge 12, and we're going to pop that in place of our current one. That's yeah.
Very nice.
And of course, different resonator and different, like our duplexer for our NMR needs to be tuned to a different range, but we built all that ourselves. So yeah.
Okay. Well, many congratulations. It's terrific work.
Thanks.
So John, looks like you're very popular. Here's another question. Do you think that it's possible to do spatially resolved measurements with this technique? And that's by Finn Sambrutsky.
Well, it depends on what you mean by spatially resolved, right? So in the one case, there sort of is a kind of spatial resolution because, for example, when we take a protein, we put a spin label at one particular location on the surface of the protein. So there's this sort of like nanoscale resolution. If you're talking about doing this with a material where, for example, you couple this with like a traditional NMR diffusion measurement or something like this, absolutely, right? They've already Uber et al. Did this with and actually and Stops Lab also, there is precedent for doing that basically. Yeah.
Thank you. I was more thinking about when you would have a concentration gradient, for example, in your NMR tube. If you could measure if differences between the top and the bottom of your tube.
Oh, like a dozy? You mean kind of like scanning along the dozy dimension in an NMR case?
Well, that's what I was saying with diffusion. But yeah. You just take an image for sure. Yes.
Is that what you're saying?
I guess so.
Yeah.
Actually, I don't know. I know very little about NMR. So
yes. I mean, basically, no. So one of the nice things. Okay, so one of the nice things then about NMR is that basically anything you can think about doing with MRI, you can just add a spectral dimension to it, right? So in our context, what you would want to do is add like field gradients to give you that imaging capability, right? You can also do what I think is maybe more broadly applicable is what like Thomas was just saying, which it was also what is you can also use that. You can use that same type of physics to sort of track what is the diffusion of say a water molecule on more of the micron scale, right? So here we're looking at the diffusion on sort of the nanoscale, right? We could couple that to like the diffusion on like the micron millimeter scale with techniques that are basically already developed, right? So yeah.
Thank you a lot for that.
Thanks.
Great. Thanks for the question. Murugusen Vela Yutam asked the question, can we use this method to study the formation of biomolecular condensates inside the cell, which is probably very popular right now?
So, yes, is the short answer, right? Absolutely. And if you have, I mean, honestly, yeah, we're talking with some people who have interesting systems. If you have an interesting system, shoot me an email, like I'm happy to, like I said, yes, we're happy to do these types of things. And so I would say yes, right? The challenge with doing it actually inside of a cell is there is a general like ESR, EPR challenge there, which is that even what interesting is I learned recently, even more so E. coli rather than mammalian cells have a real propensity for going and attacking tempo groups, right? Because they don't like the radical, right? And so there are like the EPR folks have developed some nice spin labels that are sort of resistant resistant to that process to varying degrees depending on the spin label, right?
So
yeah.
So in this case, you would switch to a different radical. I mean, right now we only talked about nitroxides all the time. I mean, you could easily
Well, I mean,
use
typically
other radical.
it's still a nitroxide, right? But typically you have this tetramethyl group and you modify that in some way to protect the nitroxide group.
Right. But your method you could do with any other radical. It doesn't have.
Yeah.
So
yeah.
Whatever. Cool. Any other questions since we have John here? Okay. Wait, I had a question. Darn it. Got too carried away. Never mind. Oh yeah. Hey, I have a question just for curiosity, and that is truly curiosity driven. There's no application. If I have lyophilized protein, did you ever lyophilize a protein, like make dry powder as good as it gets, and you shove it in the magnet? How much water motion do you still have?
I mean, I don't know offhand. Right?
I'm just curious, but there should
yeah.
be, I mean, there's water still there, right? I mean, there's no way it's all gone. So I'm just curious if there is actually still motion, if it's this lyophilized peptide.
Honestly, I mean, so specifically lyophilized peptide,
or protein.
I don't have a sense for how much residual water there is, if I'm being honest, right? You know, but you know, yeah, there's going to be some amount of motion. I mean, like actually, like in this like kind of classic review on water motion by like Ball has this really nice. It's old, right? But it's a nice, you know, kind of just big picture view of like, oh, the importance of water in biology, right? And in there, one of the things that he talks about is that like, oh, you know, like at what point, you know, stripping off the water, at what point does the protein become non-functional, basically, right? And actually what's interesting is the protein can be functional with a couple layers of water, right? You know, but yeah, obviously once you strip it all off, then it's just dead, right? And so, yeah.
Right. Nice. I thought it would be kind of cool to like maybe
no,
get
that's
like
an
a protein,
interesting to see if
put
we
a
can
label
still
in there,
see,
and then use it like a water trap. I mean, wouldn't that be cool? That would be so cool. Anyways, but it would be just fun.
No, there's also, I mean, also that makes me think of, and yet actually I think if you really wanted to answer that question, I mean, there's also these studies that they do in like solid state NMR where they read out essentially like they basically look at the effect of the water on like the relaxation of the, you know, of the spins nearby, right? And they use that to kind of get a sense of how the water's moving, right? And actually, I would not be surprised if, you know, actually there was something very similar that had been done in that realm, right?
Right. Well, it would be cool. I mean, I don't know. I would have a lot of fun with that.
Yeah.
Anyways, all right. Well, anything else, guys? Please ask away. If not, we would thank our speaker. All right. Well, thank you, John, for this fascinating talk. It was actually really fun.
Thank you all very much. It was very enjoyable. So yeah.
Thank you.
Okay.
I hope to see you soon. All right.
Okay. Take care all.
Thank you, John. Bye, John. Bye, everybody.